In the United States, major depressive disorder affects roughly 48 million adults. Managing a public health burden of this scale has historically relied on generalized solutions. The first line of defense is usually systemic pharmacotherapy. However, a significant subset of this population fails to achieve adequate improvement through traditional antidepressant medications alone. For those who don't respond to medication, standard transcranial magnetic stimulation, or TMS, is an option, but it is clinic-bound. The standard protocol requires patients to commit to daily in-office visits for 6 consecutive weeks. Standard TMS also faces an anatomical limitation. It relies on generalized scalp measurements to position the magnetic coil, rather than mapping the specific, unique structure of an individual's brain. This leaves psychiatric care trapped between two extremes: biochemical treatments that are scalable but often ineffective, and clinical neuromodulation that is effective but logistically restrictive. The recent FDA clearances of the SAINT Neuromodulation System and the Flow FL100 device mark a structural pivot in how clinicians approach depression. These devices address two separate failures in the current system: the need for rapid stabilization in acute severe cases, and the need for massive population-wide access to care. We can visualize two diverging clinical strategies. Branch A, defined by the SAINT system, optimizes for anatomical precision and rapid treatment within a hospital. Branch B, defined by the Flow FL100, prioritizes scalability and patient compliance by moving treatment into the home. Addressing the depression epidemic requires moving away from a one-size-fits-all model. Instead, psychiatry is building specialized tools for both ends of the acuity spectrum. The targeting problem in standard TMS is caused by biological variance. The exact location of the dorsolateral prefrontal cortex varies between individuals by several millimeters. While standard TMS assumes anatomical uniformity, SAINT utilizes structural MRI paired with resting state functional connectivity MRI to find the target. Clinicians upload these scans to specialized cloud software. This software calculates neuronavigational coordinates, allowing clinicians to position the TMS coil over the exact treatment target. This represents a shift toward individualized, circuit-level spatial targeting, removing the anatomical guesswork that limited previous neuromodulation protocols. The SAINT system delivers treatment using intermittent theta burst stimulation, or ITBS, which uses magnetic pulses to modulate brain activity. The protocol is highly accelerated. Instead of weeks, patients receive 50 treatments over just 5 days, administered as 10 treatments per day with 50-minute resting intervals. In clinical trials, this protocol achieved a 79% remission rate on the HDRS-17 scale. The speed of these results was a primary finding. The average time to remission was approximately 2.6 days. Data also indicated a rapid elimination of suicidal ideation at the 1-month mark, as measured by the CSSRS scale. This 5-day schedule enables a new level of clinical capability to stabilize acute, hospitalized, or highly treatment-resistant patients. Despite its efficacy, fMRI-guided TMS is logistically and financially unscalable for a population of 48 million. Most patients cannot access high-cost hospital-based imaging. This accessibility gap is where the Flow FL100 fits in. It is an FDA-cleared Class 3 medical device designed for home use. Unlike TMS, which uses magnets, this device uses transcranial direct current stimulation. It delivers a constant, low-intensity 2.0 milliamp current to brain regions involved in mood. The headset uses single-use cellulose sponges soaked in saline. These electrodes lower skin impedance, ensuring the electrical signal is both strong and uniform. To maintain safety and compliance outside the clinic, a Bluetooth-enabled smartphone app governs the headset, preventing the patient from exceeding daily or weekly usage limits. By shifting hardware from the clinic to the home, the device removes the primary barriers to neuromodulation, the cost and time required for daily appointments. Clinical skepticism has traditionally surrounded at-home neuromodulation, specifically regarding whether patients could safely and effectively self-administer the treatment. The EMPOWER trial addressed this by testing 174 subjects in a double-blind, randomized, sham-controlled study conducted entirely remotely. The study was structured in 2 parts: a 10-week blinded phase followed by a 10-week open-label phase. To prevent overstimulation, the trial used a tapering schedule: 5 sessions per week for the first 3 weeks, then 3 sessions per week for the remainder. The primary endpoint was successful. Patients using the active device showed a statistically significant reduction in HDRS-17 scores compared to the sham group. At the 10-week mark, 58% of the patients in the active group achieved clinical remission. Safety data was similarly positive. The self-reported adverse event rate was 4.5%, with side effects primarily limited to local skin irritation and headache. These trial results proved that clinical-grade efficacy and safety monitoring can be achieved in a domestic setting. The dual clearance of these devices provides clinicians with a circuit-based toolset for different levels of patient need. Mapping these interventions reveals two modern treatment paths. SAINT occupies the top left quadrant, offering the fastest remission but requiring high clinical resource cost. Flow FL100 occupies the bottom right. Remission takes longer, but cost and complexity are significantly lower. This creates two distinct clinical funnels. Severe, refractory patients who require rapid stabilization must be routed to the 5-day SAINT protocol. Patients who are failing their first medication can now substitute to high cost of standard TMS for the remotely monitored flow intervention. Psychiatry is transitioning away from trial-and-error generalized biochemical management. The data now supports a move toward targeted, precision electrostructural modulation.